Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, a biochemistry faculty. In this video, we are going to discuss the chemistry of the amino acids and the structural organization of proteins. Basically, in this we are going to discuss that how these amino acids are going to form the proteins, how a polypeptide chain is produced and what is the structural organization there. Okay, so I would name the topic as structural organization of proteins. Now see, we have already discussed about what are the amino acids, 20 standard amino acids, what are their properties. The most important property in amino acids which we have to understand is the isomerism, and in isomerism we always take alanine as the reference molecule because there is D-alanine, there is L-alanine, right? And then another thing is isoelectric point. We should know the concept of isoelectric point, which means that whenever we are talking about any amino acid, it will be separated at that pi. And what is that isoelectric point? When the net charge on any protein is or an amino acid is 0. Because amino acids have a tendency to form amphoteric substances, means they can donate a proton, they can accept a proton as they have one amino group and they have one carboxylic group. Okay, now the question here is, in this structural organization of proteins, how one amino acid combines with another amino acid, what kind of bond is there and how everything is organized, that is what we are going to discuss in this video. So, the generalized structure of amino acid, we know it is having NH2 here, COOH and R group. So, this is the general formula of all the 20 standard amino acids. Now, the question is how a protein is produced or a peptide bond is produced. So, what is a peptide bond? Because always remember, this peptide bond is considered to be the strongest bond. It is the strongest bond. Why it is the strongest bond? Because it is resonance stabilized. Very important. Okay. So, those bonds which are resonance stabilized, they are obviously strong in nature. Then coming over to this, how this peptide bond is produced. So, always remember, one amino acid will combine with another amino acid. So, if I say this is the structure of one amino acid, I can write NH2 like this, COOH, and here R group is there. This amino acid, when it combines with another amino acid, H, NH2, COOH and R. So, we can say that simple water molecule will be lost like this, minus water. And what we get is, we get this type of structure, CH, R, NH, C double bond O, CH, NH2, R, COOH. So, this becomes the peptide bond. Okay, and this was actually discovered by Sir Ramachandran also, and he gave a concept of Ramachandran angles. They were known as Ramachandran angles. Psi and phi. And for a fully stretched polypeptide, it is 180 degrees. So, if we talk about this, then another, if third amino acid will come, again a peptide bond will be produced. So, this is known as a dipeptide. And this is how we can say di, tri and a polypeptide is produced like this. So, a polypeptide is having the amino acids N. So, this becomes a polypeptide. Now, what are the biologically important peptides first of all, and how they are organized to give a stability to the particular structure? Because again, if we talk about the significance of proteins, there are proteins play a very, very important role in the body, in a sense that enzymes are proteins, immunoglobulins are proteins, transport proteins are there, plasma proteins are there. So proteins play a very important role in our lives and in the body. Okay, so now if we talk about this, what are the biologically important peptides? Important peptides. Now, the first one we will start up with dipeptide. And the example of this dipeptide is aspartame. Example is aspartame. Or what is this aspartame? Basically, it is a sweetener, artificial sweetener. Okay, and this aspartame is actually, we know those sugar-free tablets which are there in the market. Those sugar-free tablets consist of this aspartame, and it consists of 2 amino acids, phenylalanine and aspartate. Right. So now is what happens is, since it is a dipeptide, but yes, it is contraindicated in a person who is suffering from phenylketonuria. Why? Because phenylalanine cannot be metabolized. But yes, it is aram se used as an artificial sweetener in the formation, in making candies, or you can say it is a again good source of sweet for the diabetic patients also. So this is a dipeptide, or if we talk about the importance, we can say, yeah, I would say ki not suggested for person suffering from phenylketonuria. Very important, right? This is the example of a dipeptide. Then if we talk about a tripeptide, tripeptide means there are 3 amino acids which will form a tripeptide, and the example of tripeptides, very, very important, one example is glutathione. And this glutathione is formed of 3 amino acids, and the mnemonic is GCG, which stands for glutamate plus cysteine plus glycine. And this cysteine has sulfhydryl group, and due to the presence of this sulfhydryl group, glutathione is an antioxidant and acts as a very important reducing agent. Right. So this is a very important point about glutathione, and it has a tendency to form pseudopeptide. So pseudopeptide is formed. Right. And then another important feature about this glutathione is it helps in maintenance as an antioxidant, helps in maintaining integrity of the cell membrane of RBCs, of the cell membrane of RBCs. And basically what happens is, this glutathione exists in 2 forms. This is the oxidized form and this is the reduced form. So this oxidized can be converted into reduced with the help of NADPH, right, which is coming from HMP shunt. And the name of the enzyme here is glutathione reductase. And this glutathione reductase requires B2 for its function, riboflavin. And when it is in its reduced form, whenever glutathione is in its reduced form, it will break H2O2 into water and oxygen, like this. And here the name of the enzyme is glutathione peroxidase. So you can see glutathione is occurring in the both enzymes, peroxidase. And this requires selenium as a cofactor. Okay, so this is the function of glutathione, that it will maintain the integrity of the RBC's membrane. Suppose if I say that glutathione is not there, that means this enzyme will be blocked. If glutathione is not working, and this enzyme will be blocked. So it will lead to liberation of the peroxide free radicals. So basically glutathione, the function is that glutathione is not letting these free radicals to form. So that is why its function is as an antioxidant. Okay, so this is a very important function of glutathione, working as an antioxidant. Then another important feature of glutathione is it helps in the transportation of the amino acids, absorption and transport of amino acids. Right, and except proline by Miester cycle. So these are the important features to remember about glutathione. Then second example of a tripeptide is creatine. Now if we talk about creatine, creatine is also, if we talk about the functions, creatine is the energy reservoir in the muscles. It's energy reservoir of the muscles, or in the muscles we can say. And it is formed of 3 amino acids. It's a tripeptide again. And what are those 3 amino acids? The mnemonic is GAM or JAM. Means glycine, arginine and methionine. Right. Now what happens is the synthesis of creatine takes place in 2 organs. First is glycine combines with arginine in the kidneys to form guanidoacetate. Right. So glycine will combine with arginine forming guanidoacetate, and this guanidoacetate will combine with methionine. This reaction occurs in the liver to form creatine. Right. So we can say that 2 organs involve the synthesis of creatine, and once creatine is produced, this creatine undergoes phosphorylation. For phosphorylation, ATP is required and it forms creatine phosphate. Creatine phosphate is produced, and the name of the enzyme here is CPK, creatine phosphokinase. And this creatine, when it loses a molecule of water, it forms creatinine. And what is creatinine? It is an anhydride of creatine. And this creatinine clearance test is a very important test for renal function. Creatinine clearance test. It's a very important component of RFTs or renal function test, whether our kidneys are working fine or not. Renal function test. And this reaction, conversion of creatine to creatine phosphate, is known as Lohmann's reaction. And if we talk about the significance of this CPK2MB, CPK2MB is the most reliable indicator reliable and early indicator of MI. Very, very important. So these are the tripeptides. You can say creatine and glutathione. And if we talk about polypeptides, so we can say that insulin is there. So many are there. Insulin, glucagon, these are the peptide hormones. So this is how the synthesis of the peptide bond takes place. Now our topic is structural organization of proteins, because what happens is if one amino acid is combining with another amino acid, losing a molecule of water, so there are interactions between R groups. So that depends whether they are acidic, whether they are basic. So we can say that whatever steric interactions are there, they will try to go in a stable conformation, and that is what is the significance of structural organization of proteins, because every molecule tends to go in a stable conformation. So if we talk about the structural organization, there are 4 levels. There are 4 levels: primary, secondary, tertiary and quaternary. Right, so these are the 4 levels: primary, secondary, tertiary and quaternary. Tertiary is considered to be the most stable one. Right, now let's take these one by one. So if we talk about primary में there is only one bond which exists, or primary depicts the linear sequence of amino acids in a protein. Depicts the linear sequence of amino acids in a protein. So obviously they reflect only one bond, or which is known as peptide bond. So the bond which is present in primary structure is the peptide bond, and I told you peptide bond is considered to be the stable bond. Okay, and if we talk about the sequence, we can simply say that like glycine, alanine, leucine, valine. So only one bond is there, which is peptide bond, right? And again peptide bond is the strongest bond. So primary में linear sequence of amino acids is there in a polypeptide, reflecting only the peptide bond. Then second is secondary structure. Now secondary structure means there is a change in the hierarchy, means the bonds are increasing. So I can say that this secondary structure exists in 2 conformations. One is alpha helix and second is beta pleated. And obviously the bonds will change. In secondary structure, the bonds will be peptide bond. Automatically by default peptide bond will be there because it is present in all the proteins. Along with peptide bond, they have hydrogen bonds. And they have disulfide bonds. Right. Ab now see alpha helix is like this. Depends upon right-handed or it is left-handed. But if we talk about this, right-handed is most stable. Most stable one. And beta pleated is further of 2 types. Parallel and anti-parallel. Right. And if we talk about parallel or anti-parallel, we can say that the bonds will be like this, like this. And these bonds are intermolecular hydrogen bonds. hydrogen bonds. And along with intermolecular hydrogen bonds, they have disulfide bonds also. On the other hand, here, like this, they have intramolecular hydrogen bonds. Intramolecular hydrogen bonds, right? So we can say that secondary structure mein there are two type of structures which will come under secondary, the alpha helix or the beta pleated. So beta pleated has parallel and anti-parallel, whereas alpha helix has right-handed or left-handed helix. Now what are the important points about this? The most important point here is proline is never found in alpha helix. What is the reason behind this? It destabilizes the helix. The helix. And another point is glycine, if present, will form bends, because glycine is also the simplest amino acid or optically inactive. So glycine if present, it will be going to form bends, right? And if we talk about the application of practical application is curling and straightening of hair. During curling and straightening of hair, this secondary structure is actually altered because the disulfide bonds are present. So disulfide bonds can be removed, they can come again by the action of beta-mercaptoethanol. So this is secondary helix, right? Now next is primary, secondary, then there are certain super secondary structures. Now what are those super secondary structures? Super secondary structures. Now if we talk about super secondary structures, it's like secondary say upar, and there are only 3 super secondary structures. Now what are they? Beta alpha beta, right? Beta meander. One is beta alpha beta, second is beta meander, and third is Greek key. Clear? Now if we talk about beta alpha beta, it is clear from the word itself there is mergence of the two structures. Okay. So if we talk about the structure of beta alpha beta, it would be, say this is beta connected by alpha. So you can see the mergence. So because the concept behind this is everything tries to go in stable conformation. Just suppose if you are listening to my video, obviously you will keep on changing your position. Sometimes you will sit, sometimes you will stand. Some students have a habit of listening, just keeping the phone in their hand and listening to the lecture. Some students are like that, they will properly sit on a table and a chair and they will make notes. So what is it? It's like stability. So proteins, as I told you, proteins are required by so many important biochemical reactions. So how this stability is done? It is done by the different type of bonds, but it's like a kind of conformation which is giving them stability. So these super secondary structures will be stable when they are in this conformation. Beta meander and Greek key is like that, like this. Beta meander which are attached like this. So it resembles the Greek pottery. So they are represented like this. Okay. Then we come over to the third one, the tertiary structure. Now, if we talk about the tertiary structure, I mentioned earlier that tertiary is the most stable structure, or whenever a protein is in tertiary conformation, it is considered to be very, very stable, right? Now, talking about the number of bonds first of all, so I can say that it is the most stable structure, most stable conformation. Now, If we talk about the bonds present in tertiary, you start with the peptide bond is there. Okay, and after peptide bond will come the hydrogen bonds. Along with hydrogen bonds, the disulfide bonds. Right. Along with disulfide bonds, the most important bonds which are giving stability to this tertiary conformation are hydrophobic bonds. Very, very important. Okay. So I can say that these hydrophobic bonds are the ones which provide stability. Okay. Peptide bond, we know it is the strongest bond. Hydrogen bonds are there, then disulfide bonds are there, and the most important bonds which are giving stability to this tertiary conformation are the hydrophobic bonds. And along with this, we can say other bonds can be ionic bonds, like that, right? Covalent bonds. Now, this tertiary structure, since the proteins, a polypeptide is most stable in tertiary conformation, they can be best visualized by X-ray crystallography. Best visualized by X-ray crystallography. Okay, and this is a very important aspect of tertiary structure. Just say if I talk about example is myoglobin. Very important, myoglobin, which is the pigment present for the transport of oxygen in the muscles. It is stable in tertiary conformation. Right? And since they are having hydrophobic bonds which are giving stability to them, so it's like hydrophobic means they are scared of water. So what will happen is कि say this is the active site of the enzyme, all the hydrophobic amino acids will form a structure like this. Okay, so it forms a, means hydro means they are scared of water, so I can say hydrophobic amino acids form core inside. Okay, so this formation of the core inside will lead to formation of a domain. So domain is like a house. Just suppose if we construct a house and we open only one door, so what will happen is कि obviously from that particular door all the people will be there and everything will be clustered, we all will be staying inside. So there is a characteristic domain formation in tertiary which is absent in primary, which is absent in secondary, which is absent in quaternary, right? So this is very important. Then after tertiary, then comes the fourth one, quaternary. Now if we talk about quaternary, this is hierarchy wise, this is the topmost one. Right, topmost one, so you can say if we talk about the bonds, it will have so many bonds present in it. Now, what happens is, how do we define quaternary? There are different subunits. There are, I would say, monomers which are going to form a polymer. Okay, so I can say that oligomers or monomers are the single subunits. Right. So, a particular subunit is going to represent all the other conformations. Just say for example, if I take the hemoglobin only, this hemoglobin has 574 amino acids and this hemoglobin exists in 2 alpha and 2 beta chains. 2 alpha and 2 beta chains are there and it is what? It is stable in quaternary structure, right? and we know the function of hemoglobin, and this is the basic difference also between hemoglobin and myoglobin. Myoglobin is stable in tertiary conformation, hemoglobin is stable in quaternary conformation. Similarly, if we talk about LDH, LDH is also a tetrameric enzyme. Tetrameric means it has 4 subunits, so it's also in quaternary structure. So it's like 4 subunits it has. Depending upon, again, if it has isoenzymes, so obviously the subunits will be placed at different locations, but yes, it is a tetrameric enzyme. Similarly, CPK, it is a dimeric enzyme. It has 2 subunits. Right. So now see what happens is, जैसे suppose if I say 1, 2, 3, 4. So this is going to represent a quaternary structure. And this particular entity will have just this one. Will have tertiary, will have secondary, will have primary. And if we talk about what is the significance of these, so you can see that there is a hierarchy, change in the hierarchy. And if we talk about the biomedical significance, the biochemical significance is, just keep one thing in mind, that if we know the structure, quaternary structure, we can come down to the primary level. Okay, that is why we can say that the structure of insulin was elucidated. It was well characterized that insulin has 51 amino acids. Okay, it has alpha, it has beta chain. How many amino acids are present in a chain? Similarly, sickle cell anemia. We know sickle cell anemia mein what happens? There is a replacement between glutamic acid and valine. Where? At the beta position, at which amino acid? At the 6th amino acid. So this is what is the biochemical significance. So I can say that example can be insulin having 51 amino acids, structure elucidated. That is why first you can say recombinant humulin was also discovered. Ki in a patient who is suffering from DM, and if there is not proper synthesis of insulin, one can go to that extent ki if we know the sequence, which sequence of insulin is not being expressed in that particular person, that can be replaced. So structural organization of proteins carry a lot of weightage. Okay. Insulin, similarly we can say sickle cell anemia. So many examples are there. In sickle cell anemia, there is a change of 6th amino acid at beta position. So biochemical significance is we can know what is happening inside the structure, right? So this is the structural organization of proteins when they are formed of the amino acids, and depending upon the type of bonds, it is changed. Now the question arises, no doubt from quaternary one can easily go up to secondary level. The question is how the sequence of amino acids can be known, isn't it? Because till primary though it's easy. We can break, we can add acid, we can add alkali, different specific reagents are there. Now the question is, I told you ki from quaternary one can go to tertiary, one can go to secondary. But how to go to primary, where primary is having the strongest bond, which is peptide bond. Right. So how this peptide bond is broken, that is what is the need of the hour, because in primary sequence we can know that what is the sequence of the amino acids, and that is what is the need of the hour also. So how to break the peptide bond? First very important thing, what are the different reagents which can break the peptide bond, right? So let's see. If we talk about a peptide bond, or if we talk about the polypeptide, we know this polypeptide has what? Has C-terminal and an N-terminal. C-terminal and it will have N-terminal like this. Right? And this is what is here in between, these peptide bonds are there. Now the question is how these peptide bonds are broken. For that there was one very important reagent which was used, was Sanger's reagent. So what we can do is, this is the C-terminal and this is the N-terminal. Right. Now how we can distinguish, how we can identify. So first of all, identification at N-terminal, right, is carried, and this is also known as N-terminal residue analysis. Terminal residue analysis. Now what happens in this N-terminal residue analysis? We have to break the bonds from the N-terminal. And for this, the first and the most important method is Sanger's method. This Sanger was the first scientist who gave the structure of insulin, along with there was one more dideoxy method to check the sequencing in DNA also. But here we are talking about the amino acids. So Sanger's method is for N-terminal residue analysis, and the Sanger's reagent is DNFB. What is this DNFB? Dinitrofluorobenzene. Okay, so basically what happens is कि we treat a polypeptide. Say if I treat a polypeptide or a decapeptide, let's take it that way, with this DNFB. It will give out one amino acid, because N-terminal residue analysis, it will take one amino acid out, it will be converted into 9. Then again treated with DNFB, and this will go till we get only the amino acid. So at every step these amino acids will be liberated, and these amino acids will be identified by TLC. Identified by thin layer chromatography. Right, very important. So this is Sanger's reagent, and Sanger was the first scientist, I would say, first person who gave the structure of insulin. Structure of insulin, right? Very important. This is Sanger's reagent. And second is, and yes, one more addition we can do is that abhi to we are writing dinitrofluorobenzene, we can put fluorescent dinitrofluorobenzene also. So what happens is, whenever an amino acid is coming out, it will come with fluorescence. So we can add addition of fluorescent DNFB can be done. Right. So this is Sanger's reagent, and he was awarded a Nobel Prize also. Why? Because he was the first scientist who gave the structure of insulin, and that is why after the discovery of insulin, that humulin was prepared. And second method is Edmonds degradation. Edmonds degradation. Right? And if we talk about Edmonds reagent, it is isothiocyanate. So we can simply say that it is iso, or you can say PITC. thiosine. Again, same concept is there. Ki if we have a say decapeptide, it will be connected by this PITC and automatically 9 and like this. So these amino acids will be identified by again TLC or some other chromatographic technique, because again we know that ki amino acids, every amino acid has its own PI. So we can say that separated on the basis of the PI. On the basis of PI, right? Or isoelectric point, which we have already discussed in our video on the properties. So this is identification of N-terminal residue analysis, because you can say that this is N-terminal. Obviously, because one amino acid is coming like this and it is forming the peptide bond. And there are certain C-terminal residue analysis also, obviously, from both the sides. So if we talk about identification of C-terminal, whenever we have to identify C-terminal, always remember we add the enzymes, like enzymes. The most important enzyme is 𝗃𝗃𝗃. Right. So 𝗃𝗃𝗃 is required to check the C-terminal. So this is how the sequence of amino acids is revealed, and once the sequence of amino acid is revealed, automatically so many things will be sorted out. Okay. So this is a very important video on the structural organization of proteins. What are the biologically important peptides? Okay. Because after this, what we are going to study is, since I have told you that the generalized structure of the amino acid is this. So all the 20 standard amino acids are going to have the same structure. So everything depends on this. And proteins are the only macromolecules in the body which have nitrogen in them. So this metabolism is studied again under two subheadings. One is as per nitrogen skeleton, and one is as per carbon skeleton. Very important. So if we talk about as per nitrogen skeleton, they undergo three processes, which are, first of all, there is transfer of an amino group. So transamination is there. Once the amino group is transferred, it is removed by deamination. And then whatever amino group is removed, it is going to form urea and it will be expelled from the body. And if we talk about as per carbon skeleton, they are of two types, glucogenic, ketogenic. Right? Glucogenic means they will give rise to glucose. Ketogenic means they are going to give rise to some ketone bodies. Okay. So this is what is the entire video on the organization of proteins. And then in the next video, we are going to study what will be the metabolism as per carbon skeleton and what will be the metabolism as per carbon skeleton. I hope you have understood this video. Thank you so much.